A very good evening to everyone. A very warm welcome to Dr. A.K. Sharma sir, all the participants, and our worthy resource persons for the day. Today is the second day of the 5-day faculty development program for our science educators from classes 6 to 10. It aims at enhancing teaching effectiveness by designing the resources around the latest theories and pedagogies. Such programs are integral to the continuous growth and success of teachers and has a positive impact on their students. The current era demands adaptive and innovative teaching methods. So without any more delays, let's start today's session with the blessings of Lord Ganesha, who is widely revered as the remover of obstacles and thought to bring good luck, the patron of arts and sciences, and the Deva of intellect and wisdom.
Jai Ganesh, Jai Ganesh, Jai Ganesh प्यारे जय गणेश जय गणेश प्यारे एक विशाल शोभित उज्ज्वल मोतियन की कंठमाल नैनायत नारे जय गणेश प्यारे सुर ज्ञान राम तुलसीदास करत नमन चरण में तिहारे जय गणेश प्यारे जय गणेश प्यारे
A very good evening. So we should start with the first session from our first resource person, Dr. Gurpreet Kaur, PGT Biology from Bharti Vidya Bhavan, SL Public School, Amritsar, with a total teaching experience of 20 years. She has been awarded the Best Teacher Award for Rotary Club, Amritsar 2023. She is a guide teacher for National Children's Science Congress for the past 20 years and has 5 research papers in a published form. So today, Dr. Gurpreet Kaur will give her session on the topic reproduction in plants. So over to you, ma'am, Dr. Gurpreet Kaur.
Thank you, Inu ma'am. Good afternoon to one and all present here. As we know, every ship needs a captain, and we all have Dr. A.K. Sharma ji with us. He's always there for us as a craftsman who will always try for our development training programs. As teachers' role never ends with their chapters are over. Today, I have been given an opportunity to discuss with you all on the topic of reproduction in plants. With due respect and permission of A.K. Sharma sir, shall I start?
Please, ma'am, please.
Thank you, sir.
All the best.
Thank you, sir. So we all are here to study the topic of reproduction in plants. Words mean again. Production means to produce. As it is well said that reproduction is the basic necessity of life in every living organism. Because if reproduction is not there, species will become extinct. To maintain the species, whether it is animals, birds, humans, insects, any living organism I'm talking about, reproduction is a must. In general, we can say reproduction is a process of producing the offsprings. In general, we say offsprings, but technically we should say it as progeny that are biologically or genetically similar to the parent organism. Because reproduction helps in maintenance of species. Here we all have gathered to talk about reproduction in plants. So I'll be taking related to the plants only. But before starting, to study the importance of reproduction, I just want to say something. During COVID conditions, I was in a habit of watching the documentaries, and I was just watching a documentary of white rhinos. In that documentary, breaking news was appearing that white rhinos, there are only two left, one male and one female. But breeding was not possible in them because they were too old. And after 2-3 months, again, I watched the video in which it was said that male rhino is no more. Now, only two female rhinos are left. White rhinos, I'm talking about, my dear colleagues. Only two white rhinos are left. That two female rhinos, they are technically doing something so that they can be maintained. But here lies the importance of reproduction. That is, the species will not be maintained anymore. So we are talking about plants now. We cannot survive without plants. Plants are a boon to the whole mankind, and they provide us with food, medicines, fibers, everything we need for our survival. Plants need to reproduce continuously so that the plants can be maintained on the earth. Again, the importance of same reproduction as I was talking about, that it maintains the species. If species are maintained, balance of the ecosystem is whole and maintained over there. Now, talking about reproduction, the plants can reproduce asexually as well as sexually. When we talk about asexual methods, they can be leaves, they can be roots, they can be stems. So plants need to reproduce continuously to maintain the species, and sexual reproduction, the reproductive organ in plants, is the flower. If we talk about asexual reproduction, there are various methods: budding, fragmentation, vegetative propagation, spore formation, etc. But here, one thing I want to make clear, in plants only we talk about vegetative propagation. Why do we talk about vegetative propagation in plants? Because plants have the vegetative parts, that is roots, stems, and leaves. No vegetative parts are present in humans, in animals, any other living organisms. So plants are the organisms where vegetative propagation is very important, is very simple, is very cheap method. And flowers, they are not needed in asexual reproduction. Why so? Because flowers, they are the mode of sexual reproduction. And flower is, we can say, the sexual reproductive organ of the plant. So as asexual, A means without. So this means no two different sexes are required, no two gametes are required, and the plants produced by asexual reproduction will be the clones, will be the, as such, the same to their parent organisms. And one very important thing is that the plants produced by asexual reproduction thrive very well in the stable environments. Talking about sexual reproduction, that is by the flower itself. When we talk about sexual reproduction, we ask student, what is flower? That is the modified vegetative shoot. But before switching over to sexual reproduction, we should talk about asexual reproduction. Asexual reproduction is of two types. That is natural reproduction and artificial methods of reproduction. See, plants cannot do artificial methods on their own. If we are sometimes planting a cutting of a plant, a stem, they can propagate on their own. I'm using the word propagate. This means they are propagating through vegetative paths by natural methods only. So natural methods are budding, spore formation, fragmentation, etc. Natural methods, they include, I repeat once again, self-propagation of the vegetative parts. For example, the vegetative parts, they have buds. If we are talking about the small students, they have the outgrowths, they have the swellings. But when we talk about the technical word, that is, we should explain them that it is a protuberance. So we can say there is a bud, adventitious buds present, which lead to the formation of the new plant. So budding is the formation of asexual reproduction, where a new plant is developing from an outgrowth. That outgrowth is known as a bud. For example, plants such as ginger, onion, dahlia, potato, they grow from the buds. See, I have bought a potato. Is this visible to everyone? Is this visible to everyone?
Yes, ma'am.
Yes, ma'am.
So can you see the buds?
Yes, ma'am.
So these buds, these are the eyes. And the sweet potato, shakarkandi, I couldn't brought it. But the thing is, that is a root. This is a potato. Very simply, by showing in the class itself to the students, these are the buds. This means this is an underground stem and sweet potato is an underground root. Buds are appearing on them. Very simply, in a layman language, directly showing to the students, we can tell them the difference of underground stem as well as the underground root also. The buds are appearing. We call them in our own languages, eyes aa rahi hain, aankhein nikal rahi hain. So these are the eyes appearing. From where they are appearing? They are appearing from the nodes. Nodes kahan hai? They are on the stem. So that is, they are appearing. We will cut them, sow them, and plants of the potatoes can be obtained at home itself. This activity we performed in the school itself. We asked the students to bring the potatoes. The students were very happy to bring the potatoes with eyes propagating on them, with nodes propagating on them. And these nodes, these eyes, they cut with a part of the potato and sow in the soil. And they were very happy to see the propagated potatoes. Very simple activity by which they can have the new plants through the process of budding. So we can say in potato appears a small outgrowths, which are known as the buds, which commonly are referred to as eyes, and each of these eyes, along with the portion of the potato plant, they can be planted, which will grow up like the clone. We are using the word clone again over here, my dear friends, because they are exactly the carbon copy of the parents. No two parents are involved, no two different individuals are involved, so there will be no variations. No variations means they will be the carbon copy of the parents, so clones of the original potatoes will be obtained from this. So these are certain pictures of the sweet potato. Again, we can see the buds appearing from them. And here, a picture of bryophyllum plant is also given. This we can see the bud appearing. Next method is from the bryophyllum. That is from the margins of the leaves. We talked about vegetative propagation. That is, we talked about vegetative parts. That is the roots, stems, and leaves. So roots is propagated in asparagus. Can you see this plant? Can you see this, please?
Yes, ma'am.
This is an asparagus plant. If we plant this asparagus, a small asparagus plant in a small area, it will propagate over there and it will propagate to the whole area, whether it is a small pot, whether it is a soil substratum, it will cover the whole area only and only through propagation through the roots itself. Only once we have to plant that plant with the roots, rest whole area will be covered by a natural propagation through roots. This is an asparagus plant. This one is the bryophyllum plant. Can you see the leaves of the bryophyllum plant? Medicinal value, lots and lots of medicinal value itself. Can you see this? On the margins of the leaves, this is a very heavy leaf. And if the leaf touches the ground itself, only then also the plants can grow from here. And these are the margins of the leaves. Can you see the margins? The tapering margins. From here arises the buds. See, this is the broken leaf. Yahan pe ek bud thi. There was a bud over here. It has been broken. So from the margins of the leaves appear the buds, and these, when fall on the suitable substratum for the plants is the soil, the moistened soil, properly aerated soil. They fall on the suitable substratum. By chance, the lower leaf is there, it touches, then also the plants grow. See the nature, they are growing on their own, they are propagating on their own, they are maintaining their species, they are reproducing on their own. So this is what, again by leaves, so this is natural method of vegetative propagation. So three methods of vegetative propagation we have studied: through buds, through leaves, through stems, and through roots. That is buds, and the leaves, that is the bryophyllum plant. Next method is spore formation. Spores, wherever you people are sitting, wherever I am sitting, there are thousands of spores present around us. But you will also agree with this, we cannot see them because they are not visible with the naked eye. Just we can do one activity. What I did in my class, we kept a moistened bread in the class and we kept one rusk in the class, the dry rusk. What happened within 2-3 days? The moistened bread was full of fungus. There was no fungus on the dry rusk. Very simple activity, just to show the spores. Then we took that bread, moistened bread, which had fungus in the lab. We saw that fungus under the microscope, and it was clearly seen the hyphae, the mycelium, the sporangium with spores in it. So this is what I want to say, that spores are not visible with the naked eye. They are visible under the microscope, but thousands of spores are present around us. They're covered by a covering. That covering is known as cyst. That cyst or the covering, they make the spores dormant. Dormant means they can withstand unfavorable conditions such as high temperature, low humidity. They cannot germinate under those conditions. But under favorable conditions, these spores germinate and they grow into the new plants. Very simple example is of moss and the ferns. That is the pteridophytes. On the lower side of the leaves, they have spores present in them. We can propagate those plants through them also. But the rhizopus, that is the fungus, those spores are not visible. They can be visible only under the microscope. Very well tested when moisture conditions, that is favorable conditions, they are given to them. They can germinate very, very easily. The next type of asexual reproduction is fragmentation. This is also a mode of asexual reproduction. Fragments means pieces. If we cut the pieces of the plants, for example, in Spirogyra and Iggy, that each piece is a fragment of that. That is a small portion of the parent plant when cut into 2 pieces, 3 pieces, 4, 5, so and so on, so on. How many pieces it is being cut? Every piece, every part, or we can say every fragment will form a new organism of which is exactly like the parent organism. Because again, I'm saying it is an asexual reproduction, not the sexual reproduction. So we started about budding, spore formation, fragmentation. Now coming to the artificial methods of vegetative propagation, or we can say asexual reproduction. Plants cannot do cutting on their own. Plants cannot do layering on their own. Plants cannot do grafting on their own. So artificial methods include 3 methods, that is cutting, layering, and grafting. Cutting is from the stems. That is best done in case of rose plants. But the problem is unknown people cut, just a direct cut is given on the stem, but we should give a blunt cut, but we can say a V-shaped cut, or we can say a oblique cut. Why an oblique cut is given? To increase the surface area. That is when planted in the soil, that should be properly aerated, that should be properly watered regularly. In dry soils, the propagation is not easy. So by cutting, any plant can be propagated very, very easily. Second is layering. Hibiscus can also be propagated by cutting. Bougainvillea can also be propagated by cutting. Second one is layering. See, in layering, what we have to do is, we have to make a layer on the stem surface. I'm taking a simple plant of asparagus. Here, the layering do not occur, but the layering occur in jasmine. Why I have taken the stem? Just to show that this is a weak stem. That is, it can be folded. A U-shaped bend can be given in this. This is a stem. This is of hibiscus, not of guava plant. I tried to give a bend in this. You can see the wound over here. Can you see the wound over here? Can you see the wound over here?
Yes.
Yeah. If it is bent, see, it will again break. It will break. The bend cannot be given to this stem. So layering is carried out in different plants which have weak stem, that is jasmine plant. Supposing here is the soil, here is the plant growing. We just give a U-shaped bend. We will bend this stem, give a U-shaped bend, cover it with the soil, and this is known as soil layering or the mound layering. Because it is very near to the soil, very soft stem, weak stem, so it can easily be given a U-shaped bend. From that U-shaped bend, after some time, roots will appear and the new plants can be propagated. But in case of, we can say, the air layering, such as in case of guavas, what happened? We just take out the bark of the guava plant because it is above the ground surface. It is not near the ground surface, but we do not cut the xylem and phloem. Then we cover it with the moist soil having various hay particles and other things in it. We just give supposing this is a stem we are taking, we just cover it with the moist soil, and this part of the part which is covering it, this shape, this is known as gooty, Gootte, this is known as gooty. Then we cover it with a polythene so that there is no exchange of heat and matter, we can say, so that the soil do not become dry. After some days, we can see the roots appearing from it. We can cut that plant and can plant it anywhere. As it is in the air, as it is in the atmosphere, this is also known as air layering. So, my dear friends, we should keep in mind in the soil layering, the stem is weak, but in case of air layering, it can be done in the hard stems, or we can say the trees. This is a method of grafting. In this, two things are taken. One is stalk, one is scion. See, supposing there are two varieties of mangoes. One variety is resistant to diseases. Resistant to diseases means its root portion is resistant. One variety is a very good high yielding variety. For example, nowadays new varieties of mangoes are there which can give mangoes or can give the yield after 1 year. Earlier, when we used to propagate mangoes, they used to give yield after 5 years. Let it be the same thing. One variety is having disease resistant roots. It bears mangoes after 5 years. The other variety is a very good variety, very tasty mangoes, high yield. But the problem is it is not disease resistant. If it is not disease resistant, then if the roots are susceptible to diseases, the whole plant will die. But what we want is we should have the characters of both. What we can do is we can have the root portion of the wild variety, that is, which is disease resistant, and we can have the shoot portion of the second variety, which is high yielding variety. How can it be done? We just take a stalk, that is the root portion, which is disease resistant, and give a wedge shaped cut, or T-shaped grafting can be done, or any type of grafting can be done. Then we take a scion, that is the shoot portion, which is high yielding variety portion. This stalk, that is disease resistant, and scion, that is high yielding variety, they both are tied with each other. The most important thing in this is that is the cambium should be intact. Cambium should be intact means the xylem and phloem should be intact. Why? Because the absorption part, the movement, the translocation should be there. After some time, we see the grafted portion and a new plant arising, which has both the characters, that is, it is disease resistant also, as well as it is high yielding variety also. Let's see a slide video which will be telling us about the technique of grafting. I think the video is not being shown. Just give us a minute, please. There must be some technical issues. Sorry. So we will continue further. Another is the tissue culture. Another method we can say is the tissue culture. So tissue culture is a technique, we can say, of micropropagation, where the plant cells or the organs are grown in a nutrient medium, where the micropropagation is the production of thousands of identical plants. We see tissue culture. See, I'll give a very simple example. Mint, pudina, that is there in our homes. It always comes with stem and leaves. Roots are not there. And if we try to propagate it through stem, it will never be propagated. But if we take just a small tissue of it, small tissue, maybe nodes, and we plant it, I should not use the word, we should propagate in the culture medium by giving everything to them from that tissue only, that is from the cells when they break down into callus. Tissue break down into callus, that is the cells. There is a very important property of cellular totipotency. Cellular totipotency is a property in which every cell has the ability to grow into a complete new individual. So the tissue culture is a technique in which a small tissue is taken, that is the explant is taken, that is put in a medium in a test tubes, and that is propagated under lab conditions. So first of all, the simple way is to select the mother plant. What desired quality plant we want to propagate. Its multiplication is done, rooting and acclimatization is done, and transferring is done to the new plant. See, a small plant is to be acclimatized. Acclimatized means we have to train it to adapt to the harsher conditions. A small baby is there, he or she cannot do anything. We train it. We train him slowly and slowly. Similarly, we are growing a plant from a small tissue. Cells, they are propagating into a complete new plant. So what is happening? That roots will appear, shoots will appear, because this medium, we are providing them the complete medium. That medium is being provided to them, which is the plants propagated in medium, they are to be acclimatized. First of all, they will be grown in the sands, then to the soils, and then to the harsh conditions of the crop fields. So this is how the micropropagation is done of the plants. From various parts, the micropropagation can be done. The first one is the meristem culture. Meristems, as we know, root apical meristem, shoot apical meristem. That is the meristematic cells which are on the verge of division. The cells which divide, they are the meristematic cells. So cells which are on the verge of division, that is the meristematic cells, if we take that tip, the primordia, the apical meristem or the shoot apical meristem or root apical meristem, they can propagate on their own in the plants, in the test tubes, and then acclimatized so that they can be transferred into the soil. So by this method only, disease-free plants can be produced. They can be successfully used for rapid multiplication of various plants. I want to quote an example of orchids. Orchids were very difficult to propagate, but through tissue culture and micropropagation techniques, they are very easily propagated in the biotechnological labs nowadays. Another methods are callus culture, suspension culture, embryo culture. Callus culture, the method is that is the selected plant tissue is placed in artificial growing medium until the callus is formed. What is callus? Callus is the cells from the explant taken. That is, they are transferred into a culture medium containing plant growth regulators for the induction of adventitious organs. After a few weeks, new plantlet is exposed gradually to the environmental conditions. My dear friends, here again, the new plant exposed gradually to the environmental conditions means that is to be acclimatized. This means from the test tubes, first of all, they will be grown into the pots containing the quartz sand, then to the soil, then to the crop fields. Suspension culture means we are growing the plants in liquid medium. That is, the cells or the groups of the plants, they are dispersed and allowed to grow in an aerated or sterile, we can say the liquid, which is the culture liquid. This culture liquid we use is most probably the MS medium. That is Mirashige and Skoog mediums. They contain all the antibiotics, they contain the sugars, they contain the auxins, the cytokinins. That is, we are giving them a well-prepared medium in which the plants can grow. Next is the embryo culture. Here the plants, they are directly grown from the embryo when placed in culture medium with proper nutrient in an aseptic situation. Next slide, please. Now coming to the flower. These all are the methods of asexual reproduction. Before coming to the flower, I just want to show you one thing. We talked about ginger. Is it visible? Which one is ginger? Can you please tell me which one is ginger? This one or this one? My dear colleagues, this one. Okay, what is this?
Both are appearing
Ginger?
same.
It's ginger.
And this one? Again ginger?
Turmeric.
Turmeric.
This is turmeric. Yes, the students don't know much about it, but I have
Yeah.
cut the turmeric over here. So we can show these things to the students in the class because they can be propagated vegetatively through asexual methods. So these are a must because we go on teaching and teaching. We just don't show them the things what they are being taught. So please, whatsoever the layering, the grafting we are teaching them, just tell them about the plants. Just take them to the nature and let them propagate the things. They will be very happy. Now coming to the flower, that is the reproductive structure. When we go to the class and ask them about what is flower, they just say the showy portion, the flowery, the nectar containing part, the aerated, we can say having scent. This is flower. But what is flower is? That is, flower is the reproductive organ of a plant. See, plants, they are growing. Seed is there. We sow the seed. It becomes a seedling, the sprouts we can say. From the seedling, it becomes a plantlet. Plantlet becomes a plant. Vegetative growth of the plant is there. Then comes the flowering and then comes the fruiting. So what is there in this is that flower is the reproductive organ because it contains both male and the female gametes in it. Male gametes also in the flowers, female gametes also in the flowers. So flower is the part which is great importance to humans. It has aesthetic, ornamental, social, cultural, and religious values. They are structurally divided into following parts. Number one is calyx and corolla. Calyx, the children know it as sepals, the green colored sepals, which protect the flower during the bud condition. Every one of us know the bud. This is in this shape. And this is totally covered by green colored leaves, the students say, but they are the sepals. They're protecting the bud during the flower during the bud condition. Next one is the corolla, which is very brightly colored. It is having fragrance also. Why this is having fragrance? Because it had to attract insects for pollination. Pollination is a must for reproduction, otherwise reproduction will not take place. Third one is the androecium and the fourth is the gynoecium. These are the whorls in a flower. Androecium, they are the male reproductive parts of a flower, which consist of stamen having anther and filament, and the gynoecium, they are the female reproductive part of the plant, which consist of the pistil or the carpel having three parts, that is stigma, style, and ovary. My dear friends, let us watch a video of this dissection of a flower. We are trying it from the link due to the technical issues. Just a minute. Wait a minute, please. Can you see this, please?
Yes, we can see.
Is it visible?
Mm. Yeah,
Yes,
it is
ma'am.
removing the petals from a flower.
I've shared the link in the chat box. If you want to see, you can see it from there also. So coming back to the flowers, from the link I've shared, that is for the dissection of hibiscus flower, we have come to know about that is the flowers may be unisexual also, the flowers may be bisexual also. Unisexual flowers means it may be either the male flowers or it may be either the female flowers. This means the whorls, that is the sepals and petals, will be there. These are accessory whorls. But the whorls, that is the staminate containing the male whorl or female whorl, that is the pistillate whorl, only one whorl will be there, not both the whorls. Unisexual, single sex will be present, either the male or the female. If it is having male part, so it will be a staminate part. If it is having a female part, so it will be a pistillate part. This means they will be unisexual flowers. But unisexual flowers, we know it is further monoecious or dioecious. See, in papaya plant, I heard someone saying, हमारे घर में पपीता लगा है, उस पे plants में flowers, fruits नहीं होते. Why so? Why so, my dear friends?
Because it is unisexual
It is a male plant.
male plant.
Only the female plants can have the fruits. Only they can bear the fruits. But condition is if the pollen they are coming from the male plant sidewise. Kahin pe male plant is there, only then the pollen can come. We see the cucurbits, ghia, karele and all, they all have the male flowers as well as the female flowers on one plant only, though they are separate. So they are monoecious. Mono means single, dioecious, di means two. So monoecious and dioecious plants. So they both are unisexual flowers. Bisexual flowers are the flowers which contain both male and the female reproductive organs in the same flower. As we saw in the dissection of the hibiscus flowers, the male and the female reproductive organs, they were present in the same flower. So these are the bisexual flowers. Next is the reproductive structure of a flower. Because we are talking about the male and the female parts, so we should be very much aware of the androecium and the gynoecium, that is the stamen and the pistil. The androecium and gynoecium consist of male reproductive parts and the female reproductive parts. Androecium has anthers and filaments. When we do the dissection, when we see the yellow colored pollens, that is the anthers have pollen grains in them. In this diagram, very clearly it is visible in the pollen grain is a sac-like structure that contains microsporangia. That is a sac-like structure. Microsporangia will contain pollens, that is the microspores. But one thing we should make the students aware is about the ploidy. Though some say it is a topic of +2 or the higher classes, but I think the students should know because we teach them the concept of XXXY. There we talk about 46 chromosomes in male, 46 chromosomes in female, 23 each, one each from 23 from father, 23 from mother, only then the ploidy can be maintained. Because if one chromosome or the other is less or more, ploidy changes, it changes the complement of the chromosomes also. So here also they should be very well aware that pollen grains or the microspores, they are haploid. Similarly, the egg in the, when the male gametes will be produced in the haploid, they will be haploid. Similarly, the female gamete, that is the egg, should be haploid, so that on fusion, when zygote is formed, that should be diploid. That's why a little bit of information should be provided to the students regarding the microsporangia. Very simple, because it will contain spores. Which spores? Microspores. What are that? The pollen grains. What are that? The male reproductive parts. Similarly, the filaments, they are the thread-like structures that are attached to the anther, and they keep the anther in place. Because otherwise, they are just hovering here and there. So filaments, they keep the anther in place. Then there's the microsporangia contain the spores, and the topic of microsporogenesis should be told to them, that is the meiotic division. They read mitosis and meiosis in 9th class, but it should be explained to them that the microsporangia contain microspores after meiosis, that is from 4 pollen grains, or we can say the microspores, the haploid they are formed, so that the ploidy in the individual plant can be maintained. This is the structure of gynoecium, or we can say the female reproductive part. It is the female reproductive part, contains stigma, style, and ovary. Ovary contain the ovules, which are present inside the ovary, which contain the female reproductive gamete, that is known as the egg. So pistil has 3 parts, we can say stigma, style, and ovary. Stigma is on the top of the carpel. Here we should tell them about the chemotropism also, because we are going to talk about pollen pistil interaction. Pollen pistil interaction means that is the stigma. That is, the stigma contains the chemicals which can check whether the pollen they are of same species or not. If the pollen they are of the same species, they can be given entry into the ovule to reach the egg to fertilize the egg. If they are not of the same species, they are discarded and they are no more viable. Chemotropism is very important. In general, in a layman language, we can say that there is a talk between them. So the pollen of the same species is given the entry, otherwise pollen they are discarded. So stigma contains the chemicals and check whether the pollen which is landing on them is of same species or not. Similarly, as we talked about microsporangia, there is megasporangia. Megaspores are the females and microspore are the male parts. So megasporangia and the megasporogenesis. Genesis means synthesis of megaspores. Where will they be present? In a megasporangia. So megasporangia will contain megaspores, which will have the egg, and it gets fertilized, we can say, with the male gametes, which are 2 in number. Here, one thing I want to say is that whatsoever is present over here, we can tell them about this ovules, the placenta, the seeds through a pea. The students, they can be given peas to take out the seeds from them very easily. They can be shown the placenta in it. So before fertilization, before the male gamete reaches the female gamete, the process of pollination takes place. One thing, one example, one true thing I want to quote over here. During COVID condition, there was a news that in China, honeybees were not there. And for the pollination of pears, the persons were standing on the trees and brushing the pollens on the flowers so that pollination can take place and pears can be obtained. So pollination is a very, very important step for further fertilization of the male and the female gamete. See, the life starts from embryo. What is life? Just an embryo. The male gamete will fuse with the female gamete. It will form zygote. Zygote is just a cellular mass. It will go on dividing and dividing will form embryo. Embryo will differentiate. Whether it is embryo of plants, it will differentiate into plants. Whether it is embryo of humans, it will differentiate into humans. Bird into bird, and so on, so on, so on. So pollination is a must. It is an agency. It is a process to transfer the pollen from to the stigma of the flower. So it is further of two types: self-pollination and cross-pollination. Self-pollination means either from the one flower to the same flower of the same plant, or one flower to the other flower on the same plant. So if it is on the same flower, this is autogamy, auto, similar, same. And if it is on the different plant but same flower, it is geitonogamy. And then second one is the cross pollination. This is also known as xenogamy. That is, the pollen grains, they are transferred from anther of a flower to the stigma of another flower or another plant. So this is what cross pollination. My dear colleagues, cross pollination always brings variation, because variation leads to survival, variation leads to evolution. That is, they can survive in the adverse of the conditions. So self pollination, plants have various devices for preventing self pollination and going from cross pollination, but certain plants are there where cross pollination is not possible, so they undergo self pollination only. So we can try this video. Otherwise, I will share this video in the group. I'll try to show you from my phone only. Otherwise, we will share this link in the group. Is it visible and audible? Okay, I'll show you. Is it visible?
Shoot away. Let it do its job. It's only trying to help the plant pollinate. Well, interesting question. Hey, friends, I'm sure you must have heard of it.
Is it visible?
Let us explore the reproductive world of
Yes, ma'am.
plants.
Yes, ma'am, it
Answer
is
an
visible.
evergreen question. What is pollination? Zoom in. So, what is pollination? It is the process through which plants reproduce, meaning birth, fruits, and
Let
crops.
us
But the vital question is, how does it happen? To know that, first we need to learn about the reproductive parts of the plant. And what are they? Well, all consists of both male and female reproductive parts. The male reproductive part of the flower is called a stamen. It is made up of anther and filament. The anther has a sac-like structure that contains pollen grains, which are the yellow powdery tiny particles that play a crucial role in reproduction. And below the anther is a long stem-like part called the filament.
let
On
us
the
know
other hand,
about the
the
process
female
of
part of the
pollination.
flower is called pistil, consisting of stigma and ovaries. The stigma has a glue-like substance, whereas the style is a thin stem-like structure that connects the stigma to the ovary, a sac-like structure that contains eggs which reside in ovules. Now that we have learned about a plant's reproductive parts, let us know about the process of pollination. Pollination is a process through which pollen grains are picked from an anther and transferred to a stigma. This process can occur in two ways: self-pollination and cross-pollination. So, let us look at them in detail. Self-pollination occurs when the pollen from the anther is deposited on the stigma of the same flower or another flower on the same plant through wind and water. whereas cross-pollination occurs between different plants in which pollen grains are transferred from anther of one flower or plant to the stigma of another to each bird, wind, etc. known as pollinators. For example, when a bee sits on a flower for sipping nectar, the pollen gets stuck on its body, and when it flies to the next flower for more nectar, the pollen from the bee's body gets deposited on the female reproductive part. And when that happens, the pollen gets slipped into the ovary and enters the ovule, where seeds are formed through a method called fertilization.
So here we could see the process of pollination, that is self-pollination, autogamy and geitonogamy, and cross-pollination, that is xenogamy. So there are various agents of pollination, that is abiotic and biotic. Abiotic means the non-living ones, that is the air and the water. And biotic means the living one. They can be humans, they can be animals, they can be bats, they can be birds, insects, anyone working in the fields or having nectar or any other activities with the flowers. So they have different characteristic features. Abiotic, obviously they will be light. They are to be carried over to long distances. No agency as such is there, because as the water is moving, they are moving. As the wind is moving, they are moving. So they are to be produced in large numbers. They need not to produce any nectar. They should not be fragrance. They should not be colorful, but they should be having some structures which are carrying them to long distances. That should be plentiful in number. This is the slide showing various living agencies, the biotic agencies, that is insects, that is entomophily, ornithophily, that is bird pollination, chirotelophily, that is by bats, and melecophily, that is by snails. Now coming to the pollen-pistil interactions. Pollen-pistil interactions, as I was talking about in a layman language, we can say it is a talk between the pollen and the stigma. Very easily we can tell students as if we are teaching them about hydrotropism, phototropism, geotropism, and the most important thing is the chemotropism, that is the chemicals present on the stigma which will recognize whether the pollens they are of the same species or not. If they are of the same species, they will enter the ovary, reach the ovule, and will fertilize the egg. If they're not of the same species, they will be discarded and they will be no more viable. So my dear friends, the sexual reproduction in flowering plants is also known as double fertilization. This is also known as triple fusion. Some teachers, they say we should not tell the students in details about how many cells are there, how many haploid are there, how many diploid are there, but I'm of a view we should tell them. How can we explain double fertilization? How can we tell about triple fusion? How many cells they are fusing? That's why 3 cells are fusing. That's why this is triple fusion. But if we are not going to tell about them about the cells, different type of the cells present in the ovule, how can we tell about what do you mean by double 2 fertilizations taking place? So we have to tell them about the meiosis, the 8 cells produced, how they are produced, 4 towards micropyle, 4 towards chalaza end. Now, what is micropyle? What is chalaza? Micropyle is a small opening between the 2 layers, 2 coverings, 2 outer protective layers of the ovule. Those protective layers or the coverings, they are the integuments, which will later become the coverings of the seed, that is testa and tegmen. In the micropyle, the ovule through to the ovule will reach the 2 male gametes, which are released to the pollen grains. One of the male gamete will fuse with the egg, which is towards micropylar end. One of the male gametes will fuse with the 2 polar nuclei, which are coming in the center from the 2 ends of the ovule. Those 2 polar nuclei, now they are fusing with the male gamete. 3 cells are fusing, that will be triploid in nature, and the process is triple fusion. The zygote is formed by fusion of male and female gamete. So this is what we can say, that is the male and the female gamete fuse to form the zygote. This zygote is diploid in nature. So ultimately, this will form seed. What seed is? it is having embryo and endosperm. Am I audible?
Yes, ma'am.
Okay, thank you. Thank
Yes,
you.
ma'am. Yes,
Thank you.
ma'am.
Thank you. Thank you. Thank you so much. So what is there in this is, I was talking about zygote leading to formation of embryo and primary endosperm nucleus leading to formation of endosperm. Dear friends, we should tell them, just bring a rice grain. Rice grain means just a seed or a grain of rice, and show them towards the light. When you are going to show them towards light, one will be whiter portion, one will be translucent. It contains embryo and endosperm. Very easy to teach them where embryo is, where endosperm is, because it is clearly visible in that. Very easy to tell them. Just bring rice grain from homes, show them the rice grains. One will side will have embryo, the other will have endosperm. So moving further, again, a video is there if it is working. Otherwise, I'll show you from my phone itself. This is related to the fertilization process. I'm sorry for this inconvenience, but technical issues, we cannot say anything.
Don't say that. Gold are the reason we get fruits and vegetables, and their creation is no less than a wonder. Hey friends, in today's episode, let us learn about the formation of seeds and answer a life-proving question. What is fertilization? Zoom in. Plants. They are nature's best gift to us as they provide food, clothes, and even shelter, because of which they are known as universal or primary producers. And as we know, just like other living things, plants have a life too. And just like us, they tend to respire, develop, excrete, and also reproduce. Yes, my dear friends, plants majorly reproduce through three main steps, which are pollination, fertilization, and lastly, germination. We have already learned about pollination and germination in our previous videos, which leaves us with the process of fertilization. And what is that? Fertilization can be defined as a process of sexual reproduction in plants, in which the male gametes or pollen merge with the female gametes or ovum to form a diploid zygote, or the eggs that later develop into seeds. But the vital question is, how? Well, it all starts with flowers, which are the reproductive organs of plants. Yes, as we know, during the pollination process, the pollen gets transferred to the female reproductive organs and slips inside the ovary. After which, the pollen tube opens into the ovule and bursts into the embryo sac. Here, the male nucleus unites with the egg's nucleus inside the ovule, where a zygote is formed. And how does this happen? For that, let us look inside the pollen first. Pollen grains are yellow, powdery substances that consist of two layers.
So this is the video related to, we can say, the germination, that is the fertilization.
It travels all the way
This
down
is how
to
it is
the
moving.
microphile, where one of the gametes joins the egg and forms a diploid zygote, whereas the other gamete joins the two polar nuclei to form a triploid endosperm nucleus, which will transform into a nutritive tissue and provide nourishment to the developing embryo. This process is called double fertilization. Every flowering plant undergoes a double fertilization process at least once, after which the ovary transforms into fruit.
So my dear friends, was the video related to double fertilization. Sorry for the technical issues. I will share all these links in the chat box. If you want to watch, you can have. So moving further with the slides, that is, we were talking about double fertilization. So these are the fertilization events which we had seen in the videos. That is, the one of the male gamete fuses with the polar nuclei leading to triple fusion. The other fuses with the egg leading to the formation of zygote. Zygote forms the embryo that will be diploid, and the polar nuclei on fusing with the male gamete forms the endosperm, which will be triploid in nature. So these are the post-fertilization structures, the answer to the fertilization, formation of endosperm and formation of embryo. Endosperm formation means that is, it is leading to the formation of a nutritive tissue. See, seeds are germinating in the soil. They also need nutrition. How can they germinate in a dry soil, which is totally without aeration, without water, and without nutrition? We also need nutrition to grow. Development is also required. This means some nutritive tissue. See, nature has provided wonders to them. So the nutritive tissue present in the seeds is the endosperm. So endosperm will provide the nutrition to the growing embryo. And embryo, the life, the starter of life, the starting kit for the future life, will lead to the formation of roots and shoots. हम लोग घर में करवाते हैं, हम लोग sprouts बनवाते हैं. That is the embryogen. That is, we can say, the seedling is being formed. So this is the formation of seed and fruit. Seeds, this is a monocot seed and this is a dicot seed. Seed, we eat maize, corns, that is a monocot seed. We eat kidney beans, they are the dicot seeds, 2 cotyledons. And this is a fruit which has the coverings, that is the epicarp, mesocarp, endocarp, this is the pericarp. And inside the seed will be present the embryo and the endosperm. See, some students, they bring mango seeds to school, but they bring it with endocarp. They don't, we can say, distinguish between what is endocarp and what is seed. So we planted mangoes in the school. One we planted with endocarp, one we planted after cutting the endocarp. The cut endocarp seedling that grows very well in time, while which had endocarp that took some time to grow, thus showing that this is the covering of what? This is the ovary wall, the third part, that is the endocarp, and the seed is, that is the ripened ovule, is present in between. So such small activities, when performed by the students, they are very happy to perform and they can grow plants on their own. Next, we can say this is the summarized form of sexual reproduction. That is, the life started from enthesis, pollination, pollen tube germination, that is going to the embryo sac, zygote, embryo, seed, germination, again a sporophytic plant, again the same steps. That is reproducing, producing again and again. So this is the summarized form of sexual reproduction. One thing I just want to share with you is the artificial hybridization. That is, asexual reproduction is taking place, vegetative propagation is taking place, sexual reproduction is taking place. But what we want to have new things, because the humans, they have the desire to have the best quality of the things, the food, the plants, everything should be the best. So that can be attained by artificial hybridization. That means distant hybridization by breeding methods. That can be done by emasculation, bagging, tagging, and rebag. Emasculation means removal of anther from the bisexual flower. Then the bagging is done. Bagging means that is, we are doing covering the removed anther flower. This means that is having only and only the stigma. Why we are doing bagging is because if it is not covered, if the bag is not put over it, then what will be there? Any pollen can do the cross-pollination over there. So it's better before opening of the flower, bagging should be done. So we can say emasculation should be done so that we can say after doing emasculation, covering is there. That is known as bagging. When bag is being put, we put the tag over there so that the tagging is done, where it is written, at what time it was done, why it was done, on which day it was done, emasculation is being done, what we have to do further. Then we pollinate, that is dust the pollens over it on our own. How do we dust? We can do it with the help of a brush, or we can do it with the help of a any other directly from the pollens from the plants. This we do is of our own desired plant. That is, we can say the own desired quality. After that, immediately rebagging is done. Why rebagging is done? So that any other pollen do not come and interfere with it. I again had a short video. I will send the link in the chat box if it is not working. So I'll try to open it. I'll try to play it in the phone itself. If it is not working, then I will send that video in the cell that we can say the link in the chat box. I'll send the link in the chat box so that you can see the wonderful video of emasculation, tagging, bagging in the chat box. In short, we can say the steps involved in hybridization are selection of parents, evaluation of parents, because we need to have the parents which variety we want to have. Then is the emasculation, that is removing of anthers. Then after removing of anthers, bagging is being done. See, you can see in the diagram here, in the pic here, the bags are being put over the plants where emasculation has been done. When I was young, our neighbors were there in the university. What they used to do, they had cucurbita plants in their home, ghiye ke, but they used to dust something of the small creamish color flowers. I used to ask them, what are you doing? So they used to say, that is not having pollination property over here. So we are dusting the pollens. See, knowingly, unknowingly, I remember today, they were doing pollination. They were doing hybridization. But that is not a case of artificial hybridization. We should say they were doing pollination. But in the same way, we can do hybridization, artificial hybridization. But the thing is, under very controlled conditions by emasculation, bagging, tagging, pollination, and then rebagging. Whatsoever the plants produced will be of our own desired will. So to conclude my topic today, we can say reproduction is very vital because it maintains the species. Without reproduction, a very small word is there, extinct, but a very big thing to be considered. Extinct means that it is not there now. So reproduction is very vital to ensure variation and perpetuation in organisms. Reproduction is a very crucial step in the lifespan of organisms, which ensures their perpetuation as a distinct species. The success of the process is largely influenced by two components: inherent biology of organisms and interactive ecological domain in which they grow and flourish. A very important thing, because the vegetatively or asexually reproducing plants can grow in favorable conditions in which they are growing, but others which are having variations, they can grow in distant environments. That is, they can grow and adapt to the changing environmental conditions. So we can say plants optimize reproductive success through interaction between these components of reproductive structures and ecological factors. One thing more I want to share, my dear friends, this is a video I'll share. This is a video of a homework being given to the students. You can give a very beautiful project. The students can make it as a herbarium by planting, taking the plants, by taking the parts of the plant, pasting them, and about the sexual reproduction, they can give the details. That's all for today. Thank you so much. If you want to ask anything, please. Thank you, dear friends. Thank you so much.
Thank you, ma'am.
Thank you, ma'am.
My sincere thanks to Dr. Gurpreet Kaur for her exceptional presentation. Ma'am, your insights and expertise has made a significant impact on all of us. And I'm sure that all my participants would agree with me.
Thank you, ma'am. But sorry for the glitch. I'll share all the links in the chat box. Thank you.
So now I invite our next resource person for the day,